This is a guide of how you can perform the ELISA test for the CCP2 kit. It will give you visually all the information you need to do the test, including the use of the Terra software. All the TheraTest Laboratory Incorporated kit boxes are labeled with the content of the kit, the expiration date, and the kit's lot number. Allow all of the reagents being used to warm to room temperature, which is 18 to 25 degrees Celsius, for 30 minutes. The kit contains a data sheet presenting the range of appropriate measurements that the calibrators and controls must fall within. The units and ranges change with each new lot. Prepare all necessary equipment to perform the test. They are listed below. Label a dilution tube for the positive control, negative control, and each patient specimen. Place the labeled tubes into the dilution block in the location indicated on the plate map. Add 1 milliliter of specimen diluent to the positive control, negative control, and all patient specimens. Using a new tip each time, pipette 10 microliters of controls and patient specimens to their corresponding dilution tubes. Following the addition of each sample to its corresponding dilution tube, move the dilution tube over one place to indicate that the sample dilution is completed for that specimen. This serves as a precautionary measurement to save your spot and avoid confusion when diluting further samples. Cap all samples firmly after adding specimens. Slowly invert the dilution tube rack 5 times to mix the samples. As a means of ensuring adequate mixture of the samples, watch as the fluid moves through the dilution tubes. 5 additional tubes representing the calibrators will be labeled and placed in the 5 empty slots in column 1. From top to bottom, calibrators 5, 4, 3, 2, and 1. Pipette 125 microliters of each calibrator into the labeled tubes. The calibrators are pre-diluted and should not be diluted further. Using a pair of scissors, carefully loosen and remove the caps from each sample tube. On the left is an example of a TheraTest plate frame and wells prepared to run a CCP assay. To the right is a plate setup representing the sample layout for the CCP assay. Set a timer to 30 minutes. Using the multi-channel pipette, carefully draw up 100 microliters of calibrators 5 through 1. Observe each pipette tip and ensure the fluid levels are equal throughout. Load the calibrators into their corresponding wells in the first column of the microwell plate. Using the multi-channel pipette, carefully draw up 100 microliters of the remaining samples, which are controls and patient specimens. Load the controls and patient specimens to their corresponding wells. Start the 30-minute timer. While the plates are incubating, prepare the wash buffer. Using a graduated cylinder, add 900 milliliters of deionized water and 100 milliliters of 10x concentrated wash buffer. Following the preparation of the 1x wash buffer, take the label from the concentrated 10x wash buffer and place it on the newly prepared buffer. Initial and label the buffer with the date it was created. When the 30-minute timer sounds, carefully flick the microwell plate into the sink in a swift downward motion. Make sure the strips of the wells are firmly placed in the plate frame. This will serve as a precautionary measurement to prevent the wells from falling out of the frame and into the sink. Pour 1x wash solution into the clean reservoir. Set the multi-set pipette to number 5. This will load 250 microliters with each press of the lever. Insert 8 new pipette tips firmly. With the tips in the wash buffer, Depress the multi-step pipette plunger to load the wash solution. Carefully pipette 250 microliters of 1x wash into each column. Lay the pipette down on the bench with the tips facing down and out. This will prevent any fluid backing up into the pipette and contaminating it. Decant the wash into a sink. Repeat this wash procedure 2 additional times for each plate for a total of 3 washes. Firmly and carefully tap each plate upside down into a paper towel. This is performed to remove as much excess wash as possible. Use a new reagent reservoir to load the enzyme conjugate. Pour conjugate into the reservoir to get ready to load. Use new pipette tips to load the conjugate. Set a timer for 30 minutes. Load each plate with 100 microliters of conjugate. Once completed, immediately start the 30-minute timer. The leftover conjugate may be poured back into the reagent bottle. Following the 30-minute incubation, wash the plate with the same technique as the first wash cycle. Wash each plate 3 times. Use a new reagent reservoir for the addition of the chromogen. A full plate of wells will require 10 milliliters of chromogen added to the reagent reservoir. For 2 plates of wells, add 20 milliliters of chromogen. Plan accordingly. Excess chromogen may not be poured back into the original bottle. Use new pipette tips to load the chromogen. Set a timer for 15 minutes. Start the timer as you begin to load chromogen into the first column of wells. Do not wait to start the timer until the entire plate is loaded with chromogen. Pipette 100 microliters of chromogen from the reagent reservoir into all of the wells. Try not to touch the edge of the wells with the pipette tips to avoid contamination. Never pour chromogen remaining in the reagent reservoir back into the reagent bottle. This will contaminate the chromogen in the reagent bottle. Discard any leftover chromogen. This is an example of wells containing chromogen following the 15-minute incubation period. Add stop reagent to the reservoir before the timer expires. Using new pipette tips, draw 100 microliters of stop reagent. Add 100 microliters of stop reagent to all of the wells. Tap the side of the plates gently to mix the samples in the wells. The rest of the stop reagent may be poured back into the reagent bottle. This is an example of a completed CCP assay. The samples in each well are indicated. Place the developed plate into an ELISA reader for analysis. To read a new plate on the Terra software, select File, Reading, New. Select CCP 4P from the list of assays. Select Reader, Read and Collect. The reader will take 75 to 85 seconds to read the plate. This is an example of absorbance measurements displayed on Terra software for the CCP assay. Cells A1 through E1 represent calibrators 5 through 1 respectively. Cells F1 and G1 represent positive control and negative control respectively. All the remaining wells represent patient samples. After measuring the absorbance values, select the Sample IDs tab. Select Edit On. Enter the sample IDs in the same order the samples were loaded into the microplate wells. Select Save once all sample IDs have been entered. A new screen will immediately pop up, and in the dialog box annotated below, type in the name of the test that is run, followed by the date and initials of the individual who ran the test. See example. Select OK to save the assay. This concludes the presentation provided by Theratest Laboratories in reference to performing the CCP2 ELISA assay, as well as a demonstration of the test result storage within our proprietary software, Terrace. If further clarification or assistance is required, feel free to contact our technical support department at 1-800-441-0771.